Good afternoon, everyone. Welcome to this really special SciTech lecture we're having today. It's a great day to be alive in the Commonwealth of North Carolina. We started this series 9 years ago, and in the 9 years that we have been presenting these speakers, it turns out that this is our 60th speaker of the series. And we have had 3,300 in-person guests for those speakers. And of course, most of you know that we beam this to other parts of the world so what happens here, there's no telling how many people see these wonderful speakers. The SciTech lecture is a joint venture, essentially, between the North Carolina Biological, NCBW, I guess is what I'm supposed to say, NCBW, and the MST division for SciTech. And it's been a very rewarding opportunity to do this, and I know that you're going to enjoy today's speaker. And as such, I'd like to invite Joan Shank of the W firm to introduce our speaker.
Thank you very much. It's really terrific to be here. Wow, 60 speakers and 9 years, that's quite a lot. I'm from the Wake Forest Institute for Regenerative Medicine, and North Carolina is really very fortunate to have such distinguished leaders in the field of in the broader field of biotechnology, and more specifically in technologies that contribute to the field of regenerative medicine, which is the vision to develop replacement tissues and organs to address diseased, damaged, etc., tissues and organs in the body. So thanks for enabling me to introduce this wonderful speaker here. Good to see you again. And today you're going to learn a bit more about 3D bioprinting. And Professor Rohan Sherwaker is our speaker today, as you know, and he is an associate professor in the Edward Fitz Department of Industrial and Systems Engineering, and he's also an associate faculty of the Joint Department of BME, Biomedical Engineering, at North Carolina State University. His team focuses on developing design approaches and scalable manufacturing technologies for the development of engineered tissues. This involves studying the relationships between tissue manufacturing processes and biomaterials, of which are critical when we're talking about scale-up and ultimately delivery of these replacement tissues and organs to patients, and their effects on the quality of engineered tissues. This knowledge is developed to develop new manufacturing strategies for the development of medical products in collaboration with clinicians and other engineers. His research has been supported, as you would suspect, by a number of granting federal and other agencies, such as the National Science Foundation and the Orthopedic Research and Education Foundation. Dr. Sherwaker is very prolific as an author, as a published author, with over 90 technical articles in the field, and he's also been recognized with several awards, including a National Science Foundation Career Award and an SME Outstanding Young Manufacturing Engineer Award. So I'm looking forward to your talk today, and thank you. Let's all give him a hand.
Thank you for the kind words, Joan. Right, happy Thursday. Everyone having fun? Cool. So today, over the next 45 minutes, I'm going to talk to you, really give you an overview of the tissue engineering technology and bioprinting using different examples. Some of you may be more familiar with some of these technologies than others. When I talk to you over the next 40 minutes, I'm going to keep it more like a story than a scientific research-heavy talk. But later, if you have any specific questions, I'm happy to discuss. I'm happy to answer any aspects that may lead to further questions. Here is the agenda for the next 45 minutes. I'll briefly give you a background and motivation of engineered tissues and why we need them. Then we will get into the concept of 3D bioprinting. I'll really talk about some strategies you can use to manufacture tissues of various kinds in the body. I want to give you a couple of examples focusing on orthopedics. So we'll take one organ that's in your knee joint, which is very commonly associated with injuries, and I'll show you two different ways we are trying to develop the design and manufacturing for that organ. And towards the end, I'll conclude, talk about some of the challenges and future directions in that, what kind of efforts are needed, what kinds of things we should pursue complementary to the biomedical technologies so we enable the scale-up of these organs so that we make it faster for these organs to get to clinics and patients and such. Okay. I've shown a few amphibians and rodents on the screen. All of them have some peculiar traits that we mammals or we humans do not have. Any guesses? Each one of them has an ability to generate a part of their body if it is affected. So here, what you would see is this amphibian, it has the ability to regenerate its arm over a few weeks if they lose it. The gecko, it can regenerate its tail. The African spiny mouse has an ability to regenerate its skin, a significant part of its skin. And deers, of course, shed and regrow their antlers. These are not the only animals, not only species in the animal kingdom that can do that. There are several. Unfortunately, we humans don't have that ability. So in humans, when we see conditions, if there is congenital anomalies, trauma and injuries, chronic diseases, because we don't have this ability to regenerate ourselves or part of ourselves, we have to rely on different strategies. One of the most commonly used strategies is using drugs and pharmaceuticals. And again, it works for treatment of some diseases, not so much for other conditions. There's also surgical repair approaches. It's really a matter of stitching things together and then letting the body do its own thing. And there's certain kinds of injuries as well as diseases where these approaches work. Very often, this might not be enough. There might be parts of your body that do not function correctly, and then doctors have to use implants. These could be medical devices that are external, but very often, if you are talking about critical organs, it's going to be something that is implanted into your body. And of course, you are also aware of grafts and transplants. In some cases, instead of using medical devices made of polymers and metals, surgeons would prefer to use grafts, take an organ from another animal or another human cadaver and transfer it to your body. So because we cannot regenerate ourselves, these are the different strategies that we end up using. Let me give you an example. Let's get specifically into orthopedics and diseases and injuries of the knee joint. Right? One of the most afflicted joint of the human body is the knee joint. There is sports injuries, but there is also diseases. Osteoarthritis affects a significant amount of population, not just in the US, but all over the world. ACL injuries, meniscus injuries are also very, very common. And if you look at the current approaches to treat these, the most common one right now is the knee implant. This is where the surgeon, an orthopedic surgeon, would go in, clean up the part of your worn-out cartilage, clean out some of the menisci, and then really replace this entire joint with an implant that is made out of a biocompatible polymer and a piece of metal. Over the last couple of decades, the number of knee implantation surgeries have been increasing exponentially, and it will continue to grow. There is no doubt that it is helping improve the quality of life. In some cases, where implants might not be an option, surgeons might end up using grafts. I've shown an example of what a knee meniscus graft looks like. These are typically allogenic in that they have to be taken from a cadaver and then implanted into a patient. Or for the knee joint, for osteoarthritis treatment, instead of putting a metal and a polymer like this, the surgeon would choose to take a healthy piece of osteochondral tissue, which is really a tissue that contains cartilage and bone as one complex, and then implant this into regions that might be affected. And again, those could come from cadavers, but those could also be extracted from the healthy part of your own joint. So these approaches have been around. They work fairly well. They have improved the quality of life. However, there are also shortcomings, as you can imagine. The least of your worry, of course, is walking through a TSA machine and it starts ringing and everybody starts looking at you funnily and you're not sure what happens. But when you have a metal in your body, that's certainly what can go wrong, but that's not your worst fear. When you talk about traditional orthopedic implants, they typically last about 10 to 20 years, depending on a variety of factors. There is also risk of infection, osteolysis, not only in the short term, but also in the long term. If you have an orthopedic implant, even 10 years later, there is a risk associated with infection. Certainly, there have been cases where these can break inside your body. These can dislocate in your body. And although the proportion of these incidences is small, it's still fairly likely. Similarly, on the graft side, there are also some challenges. First thing first, if you are looking for a graft that would come from a cadaver, you need a human cadaver. In addition to just having a graft, it has to be the right size, the right geometry for a patient. If you are a woman who's 5 feet in height and you cannot really get a graft from a male who is 6 feet in height, the geometry is just not going to match and work for you long term. So there's a lot of factors that have to be considered if you are getting grafts as well. And sometimes these grafts don't really integrate well into your body. There have been cases of that as well. So that is where current technology stands. And so where a lot of the research and scientific efforts have been invested is in trying to create engineered tissues. And I've shown a couple of examples for the knee meniscus and this osteochondral graft. And in essence, what you're doing is you're using a polymer and living cells. But the polymer that you use, the characteristics of those polymer are very different from the characteristics of the polymers used in current implants. Mainly, these polymers, these materials that you would use, are biodegradable in nature. So over a period of time, they are going to start breaking down in your body and being excreted without causing you any harm if it's the right material. In addition, you can also have bioactive ingredients in that. You can add factors which will stimulate added cells to grow, will help cells form their own tissue. And the whole idea is that down the line, the cells will start growing, they will start forming their own tissue. This original biomaterial scaffold structure, the skeleton that you had, is going to break down over time, and then you will end up with a living, breathing organ made out of your own cells. That is the whole philosophy and the idea of tissue engineering and making these. This is one of the famous examples. I know Wake Forest folks have been leading efforts on making a ear. This whole idea of making engineered tissues itself is not very new, come to think of it. Back in 1995, this was on the cover page of Time magazine. This was in November 1995. And this was the work that was done up in Massachusetts, in Boston, by the Vacanti brothers and their group, where they were able to use cartilage and create this ear that they implanted in the back of the mouse. And lo and behold, the ear started growing with the mouse. And this was really one of the first successful demonstration of this idea of making engineered tissues. If you think it's fiction, it is not. Fast forward to 2012, similar concepts that were demonstrated in the mouse were actually used to treat a patient. This was up in Johns Hopkins in Baltimore. This woman had lost her auricle due to cancer, and they used the same technology, the same fundamental technology that was used in this mouse, to first grow a ear for her using her own cartilage on her hand, and then eventually transplant it back to the right part of the body. And she had to go some more reconstruction surgeries to get it into better shape. But this really demonstrates the idea ear is certainly not the only organ that is being engineered. I know at Wake Forest here, there's probably more than 30 or 40 organs. Joan, is that right, the number? 40 now, yeah, organs that they have been working on. Some of them have been tested in patients. Some of them are in different stages of research and development. That is where the technology stands. Right now, there aren't really, unlike medical devices, these organs have not been commercialized yet, and that is really a gap in the technology as well as opportunities for workforce development that will, in future, these will make it commercially into patients for sure. So just with that overview, let me talk to you a little more about this concept of bioprinting and really manufacturing these organs. There are several ways to do it. So starting in the 90s, people have started developing these, looking at these technologies. Prior to mid-2000s, there were a lot of technologies that really used textiles-based processes to make these organs. The first bladder that was implanted by Dr. Atala of Wake Forest was also made using one of these technologies. But things have evolved, and in the last decade or so, the focus has been on using additive manufacturing and 3D printing processes to make a lot of these organs. There are several reasons for it. First of all, with 3D printing processes, you can actually make an organ that mimics your own size, shape, and geometry. So we can make a custom organ for each and every person using their CT scans or MRI scans. So it's not going to be a generic off-the-shelf item. We could make something designed specifically for you. In addition, the way the process works, you make things one layer at a time. If you have to make an organ that has different types of cells in the body or different geometries along the cross-section of the organ, 3D printing technologies allow you to do that. You can make a single organ out of multiple cell types with multiple mechanical properties, functional properties all throughout its geometry. Generally, how this would work is you get the patient-specific data from the doctor, so from your MRI scans or CT scans. We have the technology to process these, so take those images, stack them together, process them to create a three-dimensional version, and that version is then sent to a 3D printer, and I'm giving you an example of a 3D printer, and really one layer at a time, by engineering the right geometry coming out of these models, you can build an entire tissue and organ. I'm just showing you some examples, some microscopic images. What is key with making these engineered tissues and organs, it's not just this external shape and geometry, but it's also the microarchitecture that you need to make. When you make these polymer scaffolds, so to speak, what is very important is that they be extremely porous. If they are not porous, the cells that you would add would stick and grow nicely on the outer surface, but inside it would still remain a bulk polymer. So it's very important that your structure be extremely porous, and depending on what type of tissue you are going to make, what type of cells are involved, the pore geometry, the porosity also has to vary. Different cells would perform differently under different microarchitectural designs. So with 3D printing, not only is it that macro geometry, but you could also create different microarchitectures suited to a particular tissue or organ. So very broadly, using this process, layer by layer, you can build organs, but there's multiple ways you can do this. different organs have different kinds of requirements, and a single type of process, a single type of approach may not work. So very broadly here, I've tried to classify 3 different type of approaches, and then we'll get into 2 of those with some more examples. In the simplest approach, you make this polymer structure like you're seeing in that video clip there. You make it out of the right type of biodegradable biomaterial, and the whole idea is once you implant it into the body, the cells within your body, if it's the right material, will get attracted to the sporous scaffold. It will get into those spores. So you don't have to add any cells externally. Cells will be recruited inside your body. They will start growing. The material will degrade, and then you will end up with a living, functioning organ internally. This is one of the simplest approach in the scheme of things and may work for some organs. There is another approach. If you take this a step further, there might be parts of your body which by themselves are not very cellular, so there is not a large population of cells available that may penetrate into such a scaffold. So you cannot make a simple scaffold like this using a single biodegradable material, but you have to make a scaffold which also contains the right type of bioactive cues, and you may have to add cells separately to the scaffold, and once the cells attach to it, once they start growing, then you implant them into the body, and the body takes over. So in several cases, an approach like this might work very well in order to solve the issue. The third and the most recent approach, which is technically referred to as bioprinting, is where you do not make the structures out of polymers, but you literally take living cells, mix them in a material just like jelly. The whole concoction is called a bio ink, and then using the same type of printer, you print them directly into the shape. So you don't have to rely on your body to recruit cells like the first approach. You don't have to add cells separately to the scaffold. Here your cells are integrated throughout your construct. So as you go from your left to right, the complexity and the challenges increases, but in some sense, if done correctly, then bioprinting might lead to the most efficient solution in making a lot of different organs. So let me show you in the next 15-20 minutes a couple of examples on these extreme 2 approaches, a scaffold-based approach and a bioprinting approach to making the knee meniscus. Okay, so in my first couple of slides, I talked about this organ. This is a part of your knee joint. All of us have 4 knee menisci, 2 in each knee. These are very commonly associated with sports injuries, but also due to osteoarthritis, these can wear over time. And right now, currently, there is either surgical repair, where surgeons may come in and stitch parts of the menisci together if there are bruises, or they may have to completely take it out or replace it with a cadaver. There is no medical device or no medical product per se to solve this problem. So at NC State, in a couple of different teams, we are trying to look at what are the best strategies to make them using a polymer scaffold as well as bioprinting cells. Okay? So let me talk first about the simpler of the approaches, or the scaffold-based approach. So what I'm going to focus in the next few slides that I'm going to show you is not so much the macro geometry. As I mentioned before, you can easily make a patient-specific 3D macro geometry. What I'm going to focus on is this porosity and the microarchitecture within the scaffolds, because as you're building the organ, that microarchitecture is going to be critical to its function. What you see in this picture here is a stained image of a section of this knee menisci. And what you should notice is that the knee menisci itself, it's a very fibrous organ. It contains a lot of collagen fibers and other extracellular matrix materials. And characteristically, it is all aligned in a very particular directions. The fibers are not spread haphazardly throughout the organ. They are aligned circumferentially along the same straight line through most of the structure. And that is very, very important for this knee menisci to absorb all the stresses when you are running around or doing your daily activities. So when we try to make an engineered tissue with the microgeometry, with the porous architecture, we have to ensure that the new tissue that will be formed inside an animal is going to replicate this structural alignment in addition to the properties of the biological properties of the cells and the tissues, so to speak. So really here in this was a part of a big study where we were looking at how does this microarchitecture affect this structure and mechanics during new tissue formation. We are not at that point where we can do this in humans. We are still investigating this in animals. So what we did in this study was we know we can create this macro geometry, but when we are starting with rats, the rat menisci are about 2 millimeters in diameter. It's impossible for us to make and test things like that effectively. So we said we'll make a generic structure, a circular structure, still implanted in rats at a different location, really on its back, where there's a lot of cells available inside the body, and then see the nature, the structure of the tissue, the fibrous tissue that is formed. Remember I talked about the importance of studying the microarchitecture. So here we made scaffolds of two different designs. In one design, the size of the pore was 100 microns, so that's just about the size of your hair, a single strand of hair. And then we had some scaffolds in the second design where this was scaled up 4 times, so the pore was about 0.5 millimeter or so large. This is what these things would look like in a cross section. So we took these scaffolds of different designs, implanted them in rats. Just like in that ear mouse photo that I showed you, we allowed these scaffolds to stay inside the rats for 4 weeks or 12 weeks. So for a month, we also wanted to study how this tissue formation changes over time. So some of the rats we evaluated over 3 to 4 months. And really what we were looking for was these 4 important metrics. By the way, this was what the scaffold, a cellular scaffold, looked like when we implanted it. And when we took it out 3 months later, this is what it looked like. So you can see all the tissue that has been formed not only outside, but also inside. So there were 4 things we studied once we were able to extract these engineered tissues out. First, we checked and made sure that cells were indeed able to penetrate throughout the 3D structure. The structure is about 6 millimeter thick. It's fairly, in the scheme of things, for tissue engineering, it's a very thick structure. We wanted to make sure that the cells have proliferated throughout the scaffold. It's not just on the outer periphery, because that doesn't solve the purpose that we're looking for. In addition to the penetration of the cells, we also checked if the cells were able to form new tissue. Once we understood that, we said, okay, now let's look at the tissue. Let's look at not just the volume of the tissue formed, but let's look at that alignment that I had talked about. Are those new tissue fibers aligned in a single direction like you would see in a knee meniscus or an ACL or a tendon for that matter? And finally, we said structurally, if it looks good, how about mechanically? Because when you talk about the knee joint, throughout the day, your knee joint is taking a lot of loads, right? You're continuously walking or doing activities. So not only should that engineered tissue have the same right type of cells and the structure, but also mechanically it should have the same compression elastic modulus, so to speak. So that is what we evaluated. It's just a snapshot of the results, but the main idea that I want you to get here is we had these 2 different designs, smaller pores, larger pores. What we found was the design did not affect the ability of cells to penetrate the scaffold. So even if the scaffold pores were very small, the size of your hair, cells were equally effective in getting in throughout the structure, which was really good. That was the first good news. And we were able to quantify it and show it statistically that yes, those things, there's really no difference between these different design and time points, so to speak. Next, look here. Once we ensured that the cells were able to penetrate, we wanted to see, can the cells form new tissue? Whatever you see in pink here is the new tissue that those cells had formed once they got inside the scaffold. The white lines that you see across here are actually the strands of this polymer that we use, polycaprolactone. It degrades over a period of 2 years. So within 1 month or 2 months or 3 months, we won't expect it to degrade a whole lot. And that's why you still see those white strands in between. But what we were able to conclude here also was that the cells got in, but the cells also were able to form a lot of tissue irrespective of the size of my pore. However, where we observed differences was when we started studying the alignment of this tissue. So forget how much volume of tissue was generated. If when we look at it under a polarized microscope, what we observed was that everything you see in the red in these images is the amount of tissue that was aligned in a single direction, like you see in the knee meniscus. And here we found very clear evidence that the smaller pores, the closer they were to the size of your hair, the better the aligned tissue that was formed. So essentially, this is what we call the principle of contact guidance, where the strands of the scaffolds serve as boundaries which cells follow, and cells get their cues from those boundaries and start secreting tissue along those lines. So we have proven that not only in terms of the alignment then, we also wanted to look at the mechanical properties. And we did this a little differently than most people would do it. We did not want to test the mechanical strength of the entire bulk structure, so to speak. We only wanted to test the mechanical strength of the new tissue that was formed in between these pores. So we could not use some of the commonly used mechanical testing methods, the universal testing machines. We used what is known as an atomic force microscope. It's an instrument very commonly used in nanotechnology and nanomanufacturing research for imaging as well as processing perspectives. So we took the same machine. It's a cantilever-based probe, so to speak. You can indent the probe in a very small area. The tip of the probe is like 5 nanometers. So think it's more than 10 times smaller than the diameter of your hair. So it's a very sharp tip. You can indent the tip in an area of interest. So this is our scaffold strands, and this is actually the tissue. So you can see how small this cantilever and the tip is. And based on the resistance that the tip encounters, you can interpolate how strong or how stiff that new tissue is. And here again, we found evidence that the scaffolds with the smaller pores, which had resulted in more aligned tissue, also possess the highest mechanical strength or the compressive modulus. So essentially, although the total volume of cells and new tissue that was formed was the same, in terms of the structure and the mechanics, the 100 microns design, the microarchitecture, resulted in a better engineered tissue, so to speak. And the values that we got from this tissue were within the range that is found in a normal human meniscus or even cartilage that we tested. So the idea being, we now have the proof that the scaffolds can do this contact guidance, and now we can start designing very specific tissues for very specific organs based on their mechanical properties and their alignment. So in some tissues, that alignment that I talked about goes in a single direction throughout its 3D structures. In some tissues, it goes in one direction for a part of the thickness and then another direction for a part of the thickness. So now we are getting more into very, very specific, menisci-specific structures and mechanics and such with this proof. But that kind of gives you a flavor for how you could utilize simple biodegradable polymer scaffolds that are 3D printed to induce the meniscus. Next, I'm going to talk to you about the same meniscus, but we made it using a very different approach. We said we will not make a scaffold, we wanted to see if we could take living cells. These were stem cells. Believe it or not, these stem cells were extracted from fat tissue that is thrown away after liposuction. We don't have a whole lot of shortage of fat tissue around. But we are able to extract these stem cells, and you can cause these stem cells, if you manufacture them correctly and use the right materials, to differentiate or change into whatever tissue type you would like. So in this study, we were trying to create a new type of bio-ink, and I'll explain to you in a minute, and then print this meniscus. There are living cells within this, and then we grew this out over 2 months to see if the cells survived, if they started forming the new extracellular matrix and such. So this bio-ink that we created, earlier I was telling you, this is pretty much like Jell-O. It's the consistency of Jell-O, these materials that you get. The base material we used is what is known as sodium alginate. It's extracted from seaweed. And where alginate is used outside of tissue engineering is in cooking as a thickening agent. So we used that same basic material. We had these live adipose-derived stem cells, but we also added nanofibers to this bio-ink because orthopedic tissues like the meniscus, like you saw a few slides before, are very fibrous in nature. Their fibers are aligned in a certain way. This sodium alginate hydrogel by itself does not have those kinds of fibers, so we had to add them separately, and we created this concoction. On the meniscus side itself, this is where the 3D printing aspect helps. We actually got a scan, an MRI scan of a soccer player who had injured their knee, and through our modeling techniques, we were able to extract it out and then essentially bioprint the whole thing. And then we studied, did the cells survive the process? Because think about it, this is a manufacturing process. You have living cells that you're applying extrusion, air pressure to, and causing them to deposit in a certain way. So we wanted to see if the cells survived. These were stem cells. We added the right type of bioactive factors. We wanted to see if they started turning into cartilage or cartilaginous tissue that is found in your knee meniscus. And we got good proof that was happening. Over time, what we noticed was the cells were able to survive and keep growing. But we also noticed that the bio-inks which contained those fibrous nanofibers, the cells actually did better on them. And that was something we had hypothesized because these tissues are fibrous in nature. And we saw that through some of the other chemical assaying, essentially where you see these different colors, dots in these images, those are cells. We studied them over 2 months. I'm just showing you images at the end of 1 month. But what we started seeing was the cells actually started secreting their own extracellular matrix. That is what is really the tissue at the end of the day. We were also able to find that the cells started differentiating into chondrocytes. So whatever you see as blue in these images are the stem cells that had started off as stem cells, and now they are chondrocytes. So this was a good proof of concept. The reason you don't see a whole lot of cells in here is because when we were making this construct, we were limited by the total number of cells that we could use to prepare the bio-ink. And that, I'm going to use that idea to lead to the last part of my talk, which is really about some of the challenges and future directions associated with making engineered tissues and bioprinting. Right? So where we are as a field right now is we have established a lot of technologies for different organs. We have the ability to print different types of cells, different type of biomaterials. However, what is challenging right now is we can make one-off of these for really on the lab scale for experimental purposes. We haven't yet developed methods where we could scale these things out and make millions of these, so as to speak, so to serve patients. Or in addition to scale up, the idea of scale out, where we make hundreds of these, but each one of those 100 is customized to a patient. So not all 100 are the same. So you are making a lot of organs, but you are making each one is essentially an own organ at itself. So there are challenges, and in trying to address these, the approaches that we need to take, we really have to look at all the key stakeholders that are involved in the process. It's not the bioprinting process itself, but there is a lot of the players that I have listed here really contributing to the scale-up and scale-out issues in bioprinting and engineering tissues. So, typically I give you examples. I focus more on the bioprinting and the tissue itself. But now if you start thinking about scale-up and scale-out, what I talked about is really only the 3D printing system, and it's really only one of the processes by which you take the biomaterial or cells or you deposit it. However, this 3D printing itself sits in a very large ecosystem. So if you go one level up, the biomaterials and cells that are processed that form the input to the system, the computer-aided modeling that I talked about, that becomes an input to the system. And there is a lot of variables associated with those independent systems. That's what I said in the meniscus example. You saw those cells as some dots interspersed and not really an entire mass, because realistically, to make a living, functional knee meniscus, we would need over 100 million cells. Right now, we don't have good scale-up technologies where we could extract a small piece of biopsy from a patient from which we could get a couple of thousand cells, and very effectively, in a short time, grow them into million functional cells, which can then go into a bioprinter. So even if you have a great bioprinting machine and a process, unless you have the ability to create that raw material in the form that you need, that good process is not going to make much of a difference in the larger scheme of things. Could look at it a step further. This entire system really sits in a large supply chain, so to speak, because it's a very interesting process where the manufacturer may be bioprinting a tissue, but the source of it is a patient, and then the patient doesn't take their own cells. So the patient goes to a doctor, the doctor takes the cells are shipped to the manufacturer, the biomaterials are shipped to the manufacturer, they make it, they test the quality, all of it is sent out again, and then eventually it gets back to the patient. So it's a very large ecosystem, again, where different players are involved, and small variabilities in one step of the process can have disastrous consequence on the entire ecosystem. So one of the challenges and future directions as we try to move this forward, this field forward, is to look at tools and techniques that can complement the basic biomedical science and help the scale out of some of these things. This is just another higher level version of it. I wanted to show you how you could really look at bioprinting differently. So just to give you an idea of the magnitude of this systems level problem, if you look at this all interconnected systems, just looking at this level of ecosystem, this is what a process map would look like. All the arrows that you see are the different inputs and outputs, and you can see how things are connected between the different processes. And the bioprinting I talked about is only one of the processes that sits here. So unless you can focus and develop better methods, workforce, technologies to handle this entire ecosystem, just developing a great bioprinter is not going to make a difference in the large scheme of things as you try to scale things up. There's a lot of different pathways that we can explore to try to solve the problem. I've just put a couple of very standard tools that have been used in the manufacturing industry. They haven't been adapted very well yet in the tissue engineering space. But we have the ability to simulate systems, so not just simulate a process, but we could create an entire computer model where we can exactly show how these things different flow, and if there are changes in one single process, we can simulate and find out how it affects the rest of the ecosystem downstream and upstream, and what consequences we have. So at NC State, we are doing some work where we are trying to take these very basic high-level process maps and create these computer simulations. Right now, we are creating a very high-level simulation, but then different people from different industry, depending on their product, could then tap into that simulation and fine-tune it to their needs. What we have shown here is the high-level processes, but you could go one level deeper as well and manipulate them. But some of those tools can help you understand how you want to schedule your supply chain, how you want to lay out your processing factory, how different things, what are going to be the lead times on your biomaterials, and things going out and such. On the other end of the spectrum, you could also use what are known as Lean and Six Sigma tools. So in the automobile industry, Lean and Six Sigma manufacturing techniques, ideas have been used for the last 40, 50 years now. However, we haven't adapted them so much in the tissue engineering and regenerative medicine field. But there are several very simple tools like failure modes and effects analysis, for example. That is something the FDA expects you to do when you have a bioprocess or a medical device. But we could use the same technique and apply it to do the risk analysis in a bioprinting system. And that can give you a lot of information on how you want to organize your system and solve your problems as well. There is also issues related to measurement of quality associated with these engineered tissues. So right now, if you make an engineered tissue, the way you can tell how good it is, how functional it is you take the tissue, you cut it into pieces, you apply chemicals to it, and then you find out what you had made was really good. But what's the use if you already cut it and split it and added chemicals to it, right? Essentially, many of the techniques that are used today for quality monitoring or assessment are more lab-based techniques and destructive techniques. They can give you good information as you are developing the science, but they are not going to be very helpful when you are talking about manufacturing and producing these things. So there is also the scope to look at new non-destructive methods. One of the things we are doing at NC State is we are developing a non-destructive technique where you use electrical properties of cells to determine the quality of a tissue. So we expose our engineered tissue to a very small alternating electric field. Believe it or not, your cells have very interesting dielectric properties. If your cells are living and healthy, they will respond to that electric field differently than if your cells were dying or dead. And that will change the electric signal that we try to measure. And depending on the differences in the signal, we can kind of try to interpolate right now how good or bad the tissue is, or what is the volume of cells in a given particular engineered tissue and such. We can also use the same technique to see how the stem cells are differentiating and whether they have changed from one form to another, again, just based on the electric response to their electric field. So that is just one way. The community is also working on other non-destructive techniques as well. And eventually, over the next few years, I think it is advances in the basic biomedical science, but also these complementary approaches that will get us to a point where we start manufacturing and selling these things more commercially to benefit patients, so to speak. I want to conclude this giving you an idea of the non-clinical side of things. So far, I've been focusing on the medicine aspects and clinical aspects of making these organs to treat patients. That's not the only use for them. You could make these engineered organs for diagnostic purposes as well. Today, for the most part, pharmaceuticals are tested in rats, mice, and then sometimes they are scaled up. However, what typically happens is things that work well in a rat or a mouse may not really work effectively in a human. In most cases, they do not. And so this drug testing itself is an expensive process. It takes several years. And you cannot rely on results from a rat or a mouse to interpolate exactly what is going to happen into humans before you test them in humans. So you could make such engineered tissues. So let's say I'm developing a drug for a liver or a kidney. I create a small micro organ, a micro liver or a micro kidney, but it's made using human cells and has the same properties of a human kidney or a liver. And then I can test my drugs and do my drug screening on these in vitro models. That's a much better approach than going directly into animals like rats. So there is a company which does that. They make and sell microspheroids, and pharmaceutical companies are actually trying to use this now. They're getting into this space. Not only pharmaceuticals, but also cosmetics. So a lot of the makeup contains chemicals which can be harmful to the skin. Traditionally, this has been tested in rats and mice, pigs. But now companies are investing in engineered skin, where you can make this engineered tissue like we discussed. and test their makeup on that before they get to humans. So you could test for toxicity, you could do drug efficacy testing. You could also use these models to study different diseases. So let me give you a very basic example. Today, if you are a researcher and you want to study how cancer spreads in the liver, what you would probably have to do is inject cancer cells inside the liver of a living rat or mouse and essentially have them develop cancer and then study what happens. Well, I could create an engineered cancerous tissue. You can easily get cancerous cells using bioprinting. You can make that organ and then just study how cancer spreads using that model instead of using an animal and such. So there are a lot of these diagnostic uses as well, and as we make forays into commercialization, some of these technologies are going to be commercialized sooner than the clinical aspects in the broad scheme of things, so to speak. Diagnostics is not the only thing. I'll conclude with this slide. Food, right? There is a company, a US-based company, as well as a couple in Europe that are making burgers. You extract muscle cells from cows, so these are not vegan burgers, these are still meat burgers. But you extract muscle cells from cows, you grow muscle tissue, which is really what beef is, the burger that you eat, and then you can create it. So there's already a lot of investment and commercialization on creating food using this way. The company that makes this also has a partner. They are looking at making leather for purses and belts and such. Again, using the same technology where you don't need to kill animals, but you can create the tissue using their cells as well. So hopefully I've been able to capture the essence of where engineered tissues are going and where we could use them. And with that, I'd like to acknowledge just some of the funding that all of these studies and ideas were supported by, just mentioned earlier by Joan. And I'll open it up for some questions. Are you talking about the scaffold or the printed cells? Yes. So the scaffold that we made, we did not load it with any cells. We implanted that as is. And we studied it over 12 weeks, over 3 months. And yeah, so some of the results were at the end of 1 month and 3 months.
Any more questions?
Yes, Russ.
From your perspective, because I think it's really interesting where you're seeing things. You know, a lot of times we're deep in the weeds with Joan's group over at the Institute for Gender Medicine, and I think you brought out a very practical nature. What do you see in the next few years as the most logical, actually, biomaterial products that will come to market? If any.
I think, I don't know how qualified I am to answer because I'm really not aware of the entire space on the clinical side. From my own reading and understanding, I think a lot of products related to the bone, for example, orthopedics in general seems to be one of the fields. Of all the tissues we could grow, in the scheme of things, bone is probably one of the easiest, and it's relatively fast that we could develop it. Skin as well, engineered skin, there is already commercialization along those lines. So that is what I'll see. The other major challenges in creating more functional organs like kidneys or hearts, for example, is the vasculature. We can create bone where you don't necessarily need to build the initial vasculature in your scaffold, or cartilage, which also doesn't have a whole lot of vasculature, and you can make these scaffolds or bioprint them, and they will integrate in your body. The other organs for their function, the blood vessels, the entire network, that is just crucial, and we aren't at that point where we could effectively make it, certainly not using bioprinting. So I would see in the next few years, the development in terms of commercialization is going to happen more on these avascular tissues, so to speak.
So my part two, my question is, what do you think the workforce is going to look like for those, and you and I had lunch, so this is not a trick question for you, and Joan, please chime in as well, because this is what, happens here at Foresight Tech. So we're extremely interested in what do you think those skills are that technicians are going to have to learn.
So I think, certainly not to take anything away from even something like basic cell culture, how do you grow them? So in biotechnology, also you use cells, but in typical biotechnology processes, cells are not your products. Cells are used to make your products, so to speak. Versus here, you're using the same cells maybe, but those are your products. So the approach you take in processing them is still going to be different when you're growing them in terms of contamination and the other issues, right? So I think towards cell culture, there has to be more effort. There has to be more effort, I think, and scope in automation of things. A lot of these things, contamination can be a big issue. The lot-to-lot variability can be a big issue. When you automate things, you need to develop workforce that can work with robotics, that can develop robotics. At the end of the day, we are still going to need humans to run and operate the system. We need intelligent humans. So that is the other area. I do think some of the things that I talked about in terms of the larger manufacturing, there is already these methods and skills that have been developed. There are societies like the Society of Manufacturing Engineers, for example, that has a lot of programs. And there are medical and additive groups in those societies as well, which are trying to see how these traditional manufacturing methods can be translated as well. So I think in the large scheme of things, those would be the kinds of things. And Joan, please feel free to add. Yeah. To your point about the 25 years, right? Some of the very basic tissue engineering technologies that are coming into market, I have an example. I know somebody who used to be at Duke and then they launched their company. Very simple blood vessels, okay? Extremely simple concept, very much like that acellular scaffold on a small scale. It's been about 20, 25 years now, and they are just ready to hit the market at some point. And it's dedicated effort on a single product, a very simple product. But the amount of science you need to get these products to manufacturing, that there's a lot of money as well as efforts that go in. Oh, if there are no further questions, I'll still hang around for a few minutes if anybody wants to talk. But thank you so much for your attention and being here today.
So Rohan, we have a tradition here at SciTech to give a present to speakers. So on behalf of the college and on behalf of our faculty and staff and our administration, it's my pleasure to award you with this certificate for SciTech. And I want to thank you very much, because we had actually planned to have this talk way back in October. And just to give you a little bit of background for the people that are here, there are several people here in this audience who actually will be coming to be a part of our advisory committee for a new grant that we're going to have going forward from the NSF. And that grant looks at what we call beta skills. And beta skills are those skills that we were just discussing now. They're futuristic skills that technicians are going to have in the workforce, for the growing workforce in the future. So we're really excited to have Roanne here, and also Joan Shank, who's a part of that advisory council. And that's a national grant that we'll have imminently from the National Science Foundation. And I just want to, as well, say very much a thank you to Michael Masaglia, who's in the back, because 2 years ago, Michael and I had this idea, and this idea of going forward with beta skills, and we had to bring a group together from across the country. We did that thanks to the North Carolina Biotech Center, and subsequently now we've been told today that we actually have won that grant. So we're very excited. So Rohan, we're going to see a lot of you.